Thank you for your patience, everyone. We will be starting in about 5 minutes. Esteemed healthcare professionals, thank you for joining our webinar today on the topic of vitamin E Tocotrienols and its immune boosting potential. My name is Angeline and I will be your host for today. On behalf of SIMWO's Nutrition, it is our pleasure to welcome each and every one of you. In unprecedented times of the pandemic today, the topic of immune health has indeed garnered quite a lot of traction, We are very privileged to have an expert in immunology to enlighten us on this interesting topic today. Please allow me to introduce our keynote speaker, Prof. Dr. Ammu Radhakrishnan. Prof. Ammu is the professor in immunology at Jeffrey Chia's School of Medicine and Health Sciences at Monash University, Malaysia. Having completed her PhD in therapeutic immunology from the prestigious University of Cambridge, she completed her postdoctoral studies at the University of Oxford. Subsequently, Prof. Ammu came back to Malaysia and joined IMU in 1997 before her attachment today in Monash University. Her main area of research are in immune therapeutics in cancer, immunoregulation, as well as the use of natural compounds to enhance host immune response to cancer and other conditions. Today, she is a life member of the Malaysian Genetics Society, the Malaysian Nutrition Society, as well as a fellow of the Cambridge Commonwealth Trust and the Cambridge Philosophical Society. She is indeed an expert in immunity, and we have so much to learn from you today, Prof. Ammu, and we are very privileged to have you on board with us. Without further ado, please allow me to pass the screen to Prof. Ammu for this session.
Okay, thank you so much for the kind words. Firstly, I'd like to say thank you to Sime Darby Oils and Nutrition for giving me this opportunity to share my research. And also would like to thank all of you for coming here today to listen to my talk. So I hope you all can see my slides.
Yes, we can see them.
All right. So I'm going to talk to you today on vitamin E tocotrienols, their immune boosting potential. So as you know, I'm from Monash, and my main area of research is using natural products, in particular vitamin E from palm oil, in boosting immune responses, not just to vaccines, as well as to boost immune system towards fighting cancer. So the outline of my lecture today will be on, first of all, I'll give you an overview of the immune system, because as you know, the immune system has advanced. I mean, it's a very complex body system. A lot of new things are being founded every day, and a lot of publications coming up. So I'll just give you a brief overview, and I'm very happy to answer any questions you would have on the immune system. And then we talk about a little bit on the factors that affect the immune system. And then, as I mentioned, I am working on the nutritional aspects of the immune system. So I'll focus my research on tocotrienols, and I'll share with you some of my evidences from my PhD students' work on the vaccination model. We've done a clinical trial in humans, and as well as a similar study in mouse model, because we were trying to answer some additional questions. And then I'll share with you the results from the breast cancer mouse model. Now here, I'm not looking at the effect of the tocotrienol on the cancer. I'm actually going to share with you how tocotrienol supplementation regulated the host immune system. So if we talk about the immune system, everyone knows this function of the immune system, which is immunity, where you talk about vaccination or fighting invading pathogens like bacteria, viruses. So primary function here is, in terms of pathogen, is to prevent infections. But we know the pathogens are quite clever, and they find ways of invading your body. So to that aspect, we need to find ways to fight the pathogen. And for that, we use our immune system. If we activate our immune system correctly, then we will acquire what we call long-lasting immunity or protection against the pathogen. So this is something many people are familiar with. Another important function of the immune system is when there is injury, you need to heal. So the cells of the immune system play a role in the repair and healing processes. They help to remove old cells as well as dead cells, because you need to do this before the healing and repair can take place. Another aspect which is more of the crux of my research is the role of the immune system in identifying and destroying abnormal cells. By abnormal cells, we mean cancer cells, damaged cells, or mutated cells. So technically, your immune system is equipped to identify and remove these cells, but things can go wrong, as you'll see later on when I share with you my work from the animal work. Now, the immune system must be well regulated. If we don't regulate our immune system, we actually can end up with a number of undesirable effects. Immunodeficiency is usually a condition which is related to your genes or maybe to some therapy that we take. Sometimes it's unavoidable. Now, if you don't keep your immune system active or vigilant, doing some function, you will be more susceptible to allergy. And then if something goes wrong with the activation or the regulation of the immune system, your immune system can become your enemy. And then you have things like autoimmune disease, and that's quite a very chronic condition, and it's not good for the patient. And sometimes when patients need transplants, it is the immune system again that rejects. So these are some of the not so good effects of the immune system. But when you look at the overall picture, it actually is a very important body system. So I'm going to show in my lecture, we're going to focus on the use of tocotrienol rich fraction or vitamin E from palm oil on enhancing immune response. Here I've used a vaccine. And in terms of cancer cells, I'm just going to share with you some of my preliminary work with experimental model, a syngeneic mouse model of breast cancer. So the immune cells are called white blood cells or leukocytes. I'm sure you all know this. So they are the T cells and the B cells, NK cells. These are lymphoid lineage. And then you've got all the polymorphs, eosinophil, neutrophils, basophils, the monocytes, dendritic cells, macrophages. So they are all bone marrow derived cells because you can trace some precursor coming from the bone marrow. Now, for this lecture, I'm going to focus mainly on the T cell function and a little bit on the B cell function. And you see that this group of cell called the dendritic cell has a very important role in the activation of T cells. So the innate arm is basically a very, very simple arm. Is to prevent entry, the barrier, prevent entry. You've got skin and mucous lining, which are very important. You've got normal bacterial flora, which competes with the pathogen to prevent colonization. Then there's biochemical protection. I'm sure you all remember your complement system and cytokines, and your cells, which are basically the granulocytes, and then the monocytes and dendritic cells. So this is innate. And adaptive, it's a bit more complex. And when you want long-lasting protection, we need to activate the adaptive arm. So the adaptive arm, the key cell that you initially activate is your T helper cell. The T helper cell then helps the cytotoxic T cell to do the function. And then it helps the B cells in doing what we call class switching and developing memory cells. And all this is done by production of cytokine. Now, it's very important to realize, although yes, the textbook, they call innate and adaptive, both arms of the immune system are required. Okay, there actually is some overlap in certain functions. So it's not one alone, there's a lot of cooperation between innate and adaptive. So in order to understand when we talk about immunization, the concept of primary and secondary immune responses is quite important. So in order to understand why we go for vaccination and why we measure antibody responses, okay, what the antibody will do, okay, this is showing you what happens if you don't have antibody. The virus, for instance, your COVID-19 virus, will use its receptors to bind onto receptors on the cell, and then it gets into the cell, endocytosed into the cell, and then it releases its genetic material, and then your host cell has to now make out more viruses. And this is one of the ways where you start killing your cells. If you have antibody to the virus or some part of the virus, you can actually prevent this fusion from happening. So an antibody is easy to measure in the plasma because there are a number of kits like ELISA kits that you can use to quantify the amount of antibody. So when we talk about primary and secondary immune responses, a lot of books focus on the antibody response. So if you look at this slide is showing you this on this axis, the concentration of antibody. And in this axis is talking about time after immunization, or it could also refer to time after exposure to the pathogen. So over here is your first exposure, okay? And what you see is there is a lag phase. So please understand, during the lag phase is your innate system that is doing something to prevent all these pathogens from colonizing the host and causing all the problem. So if the innate is not able to get rid of the infection, we will be activating our adaptive at the same time. So there is a little lag phase, which is very evident in primary immune response, which is why you can't actually detect antibody immediately after the first dose of immunization. And the other thing you see is gradually you see the immune response accumulating. And the main class of antibody produced in a primary immune response is IgG. The first class of antibody produced is IgM. And then towards later on, you see IgG produced. So this means that your T cells has also been activated and promoted class switching. And what you also need to see is that the level here, it doesn't go totally down to zero, which means there's still some level of protection there. So if we were to have the same booster or second exposure to the same antigen, you'll find the response is very rapid and is much higher. And the other thing you see is the major class of antibody here will be IgG. So this tells you your immune system has been primed properly based on first and second exposure or first and second doses of the vaccine. Okay, so this is an important phenomenon when we talk about vaccination. Now, in order for the primary and secondary immune response to have this pattern, okay, you see a lower primary and a higher secondary, we must activate the T cells. In particular, the population known as the CD4 and Th1. Now, just let me explain this picture. When I started my PhD about 30 years ago, I only had to deal with two cells in terms of T cells, which was a CD8, which is known as the cytotoxic T cell, which goes and kills the target cells like virus-infected cells or cancer cells, and the CD4 cells, which make cytokine, which help to modulate some of the immune responses. But 2 or 3 years into my PhD, they said, oh, the CD4 cells, there are two types now. We call it Th1 and Th2. And both Th1 and Th2 are like the yin and the yang. One will do one function, and then the other one will inhibit the other function. So it's like an antagonistic interaction. But today, if you look at the literature, this CD4 subset has turned into many more subsets. And a very interesting cell, which is the T regulatory cells. Now, for a long time, immunologists were looking for this elusive cell. Maybe because when they were initially postulated the presence of T regulatory cell, they used to call it T suppressor cell, they were looking for a cell that expressed CD8. Well, what we find now is that Tregs do not express CD8, they express CD4. So this is again another subset of CD4 T cell. So if you look at the literature in terms of the subsets of CD4 cells, actually now there are more. So initially starting out with Th1, Th2, T regulatory cells, and now we've got Th17, 22, Th9. This is the follicular T helper cell. So it's a lot more, and we don't know if there's going to be more subsets coming in. Now, I remember I mentioned in the early part that the innate and the adaptive has a very close relationship. Now, the close relationship is via a cell called dendritic cells. Now, T cells are actually a bit unique in the sense that the T cell receptor do not directly interact with the pathogen or the antigen or the vaccine. It has to have a situation where the substance, the antigen is processed and is usually done by a dendritic cell. So antigen acquisition into the endosome, and the antigen will be processed. It means it's broken down into little peptides, and the peptides bind onto a special group of protein called MHC, the major histocompatibility complex, or in humans, it's known as the HLA proteins. So it binds onto this MHC protein, and then this dendritic cell will get activated, okay? It undergoes maturation, it undergoes activation, and then it goes to the areas where you can find naive lymphocytes. Usually the lymph nodes, you can find them in the spleen, you can find them in other lymphoid tissues. So it goes to the lymph node or the areas, other lymphoid tissues. It looks for a T cell receptor, okay, which has the ability to recognize the peptide that is presented by the MHC molecule. So you see in this example, it goes to the lymph node. It's trying to look for antigen to present that antigen to a T cell. So here it's found the T cell, so it's presenting the peptide to the T cell. And then various other signaling, and I will elaborate on this in a moment, is required before that naive T cell can get activated. Now I keep the word naive is important because the naive means that T cell has not encountered any antigen previously. So this is the first time it is encountering, and in activating a naive T cell, various conditions have to be met. Now, the dendritic cell can also be activated by other substances, like some viral protein pathogen-associated molecular patterns, inflammatory cytokines, and dead cells. So various other things. But in terms of activating our T helper cell, the peptide must be processed and presented on MHC. So it's not a simple process, okay? So why they go to the lymphoid organs and tissues is because this is the home of naive T cells and also B cells. And in order for the T cells and B cells to be activated, their receptor must engage with a particular specific antigen or peptide, and then they go through activation. Now, B cells, it's not so complicated, but T cells is a little bit more complicated. So for T cell activation, it is a complex process. It requires 3 signals which are independent. The first signal is a recognition signal, which is where the peptide, the antigen presenting cell, the APC, the antigen presenting cell, presents the peptide in the MHC protein, and this is recognized by a T cell receptor. So that's the first step, and that is showing you specificity. So if you look at this picture, this is the first step. That is your antigen presenting cell, okay? Presenting the red is your peptide. This yellow structure is representing MHC, and is presenting the peptide to the T cell receptor of a CD4 expressing T cell. So this is the first signal that goes. So it's an activation signal. Now, in order for the T cell to get activated, we must go on to signal 2, and the signal 3 must happen. If for some reason these 2 signals does not happen, then no activation will take place. So the second signal thereby is called a survival signal, where the antigen presenting cell will give a co-stimulatory signal to the T cell. So if you notice, the APC here has got other molecules. In this example, a B7 molecule binding to its ligand called CD28 on the T cell. So this then gives the second signal, which is required for survival. So for a long time, they said it's only 2 signals, signal 1, signal 2. And more recently, they've added a third signal, which is in the form of various cytokines, okay, and growth factors, which then cause the differentiation of the T cell. Now, remember, we talked about Th1, Th2, Th3. Now, the cytokines produced at this microenvironment here will drive the differentiation of the different T cells to the different subset, okay? And the particular T cell will then secrete different cytokines to modulate different types of immune responses. So all 3 steps have to happen before a naive T cell is activated. So a naive T cell, I'm just going to focus on 4 subsets of the naive T cell, and not everything, because a lot of research, a lot of information is not known about many of those. But Th1 is the main type of T cell we want to get in any immune response. Okay, now Th1 mediates cell-mediated immune response. It helps to kill intracellular pathogens like viruses. Okay, it has anti-cancer activity. And then it promotes class switching of B cells to IgG. Now, IgG is a very important class of antibody, therapeutically speaking. Now, this subset is produced in an environment where there is a lot of interferon gamma and interleukin 12. A signature cytokine for Th1 subset is interferon gamma. I alluded to you that the yin and yang, the opposite was the Th2 cells. Okay, our Th2 cells develop in an environment which is rich in interleukin-4 and interleukin-2. Now, Th2 is not the desired immune responses. Traditionally, it was important to eliminate extracellular parasites, but increasingly in developed countries, we don't actually have lots of parasites now. So if, for instance, it gets activated, you find that it's high in patients with allergy and asthma. The cytokines that this subset will produce is interleukin-4, 5, and 13. So this subset is something we do not want to encourage because it doesn't produce a lot of beneficial responses. Next, we go on to Th17. Now, Th17 is produced in an environment which is rich in TGF, transforming growth factor beta, interleukin-6, and interleukin-21, 23. It's important in autoimmune responses. So you want to suppress autoimmune responses, and it helps you to eliminate extracellular bacteria and fungi. Signature cytokine is Th17. You'll see the cytokine is called IL-17, and it can also produce IL-21 and 22. One of the other important one is your T regulatory cells. So don't think T regulatory cells are bad. Actually, T regulatory cells are very, very important. If you don't have T regulatory cells, your immune system can go haywire and start attacking cells. So one of the main functions of T regulatory cells is immune tolerance. So these are also produced in the thymus, and even in the periphery, we can induce development of T regulatory cells. So they are involved in lymphocyte homeostasis, and on the whole, they regulate immune responses. So whether you have high levels of Tregs or low levels of Tregs will determine an outcome of an immune response. So this subset develops in a condition which is rich in TGF-beta and IL-2, interleukin-2. And signature cytokines are TGF-beta, interleukin-10, and interleukin-35. So when we are talking about immune response, enhancing immune response, the two subsets that will be very, very important is the Th1 subset and the T regulatory cells. So I'll share with you how these two are related in my mouse model. So it is said that our ability to interact with the world and remain healthy largely depends on a healthy immune system. Now, actually, the COVID-19 pandemic has done a lot for people to realize how important their immune system is. Okay, so the immune system is not that difficult for us to keep it happy. There's few things that we need to remember. Now, this graph, before I forget, is to show you that there is a fine line where you get tolerance or immunity. So usually the immune system is in a dilemma. Is it going to be tolerance? Is it going to be immunity? Now, if it's tolerance, you'll find immunosuppression. If you have inflammatory responses happening, then you will have an immune response. So again, if you look, the key players, when you want to have an immune response, is your CD4 T cells, CD8 T cells, you see the interferons, okay, and all these cytokines which are more of a Th1 response. And if you look at tolerance or suppression, the key cell here you see is your T regulatory cells. Then you see TGF-beta, IL-10. So the environment, the microenvironment where we'll be looking at the balance, which is higher, which is lower, and then that determines the outcome of an immune response. Now, the factors that affect your immune system, actually there are very few factors, okay? Now, things that you cannot change are your age, okay, very difficult to change because that's something biological, and your genes, you can't change because you're born with that. Other factors I've listed here, we can change. Nutrition is one important aspect of your immune system. Moderate exercise, because if you do exercise, it has a feel-good factor, and that can stimulate your immune system. Drugs and medication, now sometimes patients are on immunosuppressive drugs, or that will suppress immune system, so they will be immunocompromised. And other factors like sleep. Now, when I tell my friends, sleep is so important, you need to do 6 to 8 hours of sleep every day, they think sleep is something is dispensable. It's not. Sleep is very important to give your immune system, immune cells a rest. Now, stress can release cortisol, which can suppress immune response. So you shouldn't be very stressed. Smoking, infections. Another area which is coming up is the gut microbiome and immune system. So today I'm just going to share with you some information on nutrition and the immune system. So if you look at the literature, it actually shows the science, the evidence shows that nutrition is a very, very important factor to support a healthy and balanced immune system. Remember, I said age is a factor which is important. There is documented evidence to show that your immune function can decrease as we age. However, we can actually reduce or retard this decline by having a healthy nutrition. So by taking in good food that supports your immune system, you can actually decline or retard this decrease in your immune function. So if you have poor nutrition, it increases your susceptibility to infection, to cancer, and you have slow healing from injury and infection. This is because when your immune system, when immune cells get activated, they need to produce new proteins. So in order to produce new proteins, you need your amino acids. So if you have a poor nutrition, this is not going to happen. So let's look at the nutrition. Now, macronutrients like carbohydrate, proteins, lipids, and fibers, I'm not going to touch that, because if you have deficiency here, it's not just your immune system that's going to be affected. A lot more body systems are going to be affected. So I'm going to just focus on micronutrients. So micronutrients, basically all the vitamins are listed here. And then if you look at the literature, some supplements like cholesterol, probiotics, antioxidants, all have been shown to have some impact on host immune system. And then some natural bioactive compounds, I've worked with some of them, not all of them, also have been shown to have immune enhancing ability. So for this talk, I'm just going to show you some evidences related to vitamin E. Now, vitamin E is a naturally occurring vitamin. Okay, the vitamin E that I've been working on is the vitamin E from palm oil, harvested from palm oil. So this is called the tocotrienol rich fraction, or generally known as TRF, tocotrienol rich fraction. Now, vitamin E exists naturally in two forms. The tocopherols, which is what many people are familiar with, tocopherols, and tocotrienols. And I also became familiar with tocotrienols when I started my research in tocotrienol about 20 years ago. So the tocotrienol rich fraction from palm oil contains about approximately 80% tocotrienols and 20% alpha tocopherol, which is the more common vitamin E that is sold in many pharmacies. Now, tocotrienols and tocopherols exist in different isoforms. Tocotrienol has actually, TRF has most of the isoforms of tocotrienol and one isoform of tocopherol. Just to tell you, the tocopherol and tocotrienol are actually structurally not very different. The next slide, I'm going to show you the structure. So this is tocotrienol structure, and this is the basic structure for tocopherol. If you notice, both have a chromonol ring and a side chain. The side chain of tocopherol is saturated. The side chain of tocotrienol is got three unsaturated bonds. Okay, and the alpha, beta, delta, and gamma all depends on the functional group that sits on R1, R2, and R3. So for instance, if R1, R2, R3 are all CH3, the methyl group, then this is an alpha tocopherol. Okay, so this exists naturally. These are natural forms. And the tocotrienols were discovered sometime in the 1960s. Okay, so from the time it was discovered to now, there has been increasing amount of research in the use of tocotrienol in various aspects of human health. Okay, started off with antioxidant, then went on to cancer, breast cancer has been the area which I've been doing a lot of studies, and then there's a lot of other effects shown. So the next slide shows you some of the many effects that tocotrienol is reported to have in human health. So anti-cancer, immune enhancement, anti-inflammation, antioxidant, neuroprotection, okay? And then others here refers to anti-cholesterol, protecting your kidney and nephroprotection and so on. And these are mainly to do with anti-cancer. So I'm going to focus on this aspect. As I mentioned, we will talk about immune enhancement and in terms of a vaccine, and immune enhancement in terms of fighting cancer. So the first study that I'm going to share with you is on enhancing immune response to vaccine. And here we're going to use tetanus toxoid vaccine. Now, why tetanus toxoid? This study was done about 10 years ago. Now, we were looking for a candidate vaccine. Tetanus toxoid was a good candidate because it's safe, there's no big adverse effects known. And all Malaysians when you're born, you would have to have had this vaccination. So, and the aim of the study was to assess the effects of palm tocotrienol supplementation on immune response following tetanus toxoid challenge in healthy female volunteers. Now, you may be wondering why we went for healthy female volunteers, because most studies will use male, but we had no choice because one of the exclusion criteria was smoking. So in that population, we couldn't find a number of males who did not smoke. And also we wanted to use a certain group of volunteers, okay, aged between 18 to 25, because there has been studies in the West which showed that there is some beneficial effect of using tocotrienol in older people. They didn't find any in younger people. So we wanted to show, actually, it doesn't matter whether you're young or old. So we wanted to use a younger population and show that we can only measure this when you use an immunological challenge. In this case, the challenge was a tetanus toxoid vaccine. The clinical study was randomized, double-blinded, placebo-controlled clinical study. So there were two test items. One was control. They received placebo for 56 days. The other group received 400 milligram of palm tocotrienol rich fraction for 56 days. And the blood was taken at 3 time point, baseline, before they started their supplementation. And then they were asked to start their supplementation after they gave blood. And 28 days later, they were asked to come back. We take the blood and then we give them the vaccine. Now, there's no need to give booster here because Malaysians, as I said, would have received this vaccine sometime as a child. And then they asked to come back at day 56 and we take another lot of blood. So from day 0 to day 56, they were either taking the placebo or the TRF, and it was double-blinded. So this shows you the results, okay? So over here is showing you day, and this is the amount of antibody, IgG class of antibody quantified. The white is the antibodies to tetanus toxoid in the TRF group. And what we did was to see whether was this antigen-specific immune response. We decided to measure interferon gamma. So we took the peripheral blood lymphocytes and grew them either in the presence of con A. Now con A or concanavalin A is a mitogen which induces proliferation of T cells. So it's a non-specific inducer of T cell proliferation. And we also stimulated them with the tetanus toxoid. So this proliferation will be antigen specific. So in both cases, you see the TRF supplementation, especially when you use antigen specific stimulation, we see a very, very high level of interferon gamma produced, which tells us that this situation is inducing more of a Th1 immune response. And this is published in the European Journal of Clinical Nutrition about 10 years ago, okay? And at that time, in that study, as I mentioned, we only had placebo and TRF. So a question was asked, what about alpha tocopherol? Because in the TRF, there is about 20% of alpha tocopherol. So we didn't want to run another clinical study. So we did a mouse study here. Again, we used the same vaccine, tetanus toxoid vaccine, challenge in a mouse model. So we tested 3 compounds: alpha tocopherol, tocotrienol rich fraction, which was similar to what was used in the human. And we used an isoform, a specific isoform of tocotrienol, delta tocotrienol. And the vehicle was the oil that was used to feed these substance. So in the mouse model, we had to give primary, secondary, and tertiary dose to boost the immune system. So after acclimatization, we start supplementing the animals with either the vehicle or different vitamin E forms. And then we gave the first dose after 14 days, second dose after 28 days, and the third dose after 42 days. And we took blood at each time point. So I like to take you through this picture. So the black dot is pre-bleed, that means your baseline. The V1 is day 28, and then the V2 is day 56. So if you look at the vehicle, you can see there is immunization has been successful because you can detect anti-tetanus, titers of anti-tetanus antibody at day 28 and day 56. This was higher in the alpha tocopherol fat animals. You see the highest response in delta tocotrienol fat animals, and you see a very moderate response, which is higher than tocopherol in the TRF fat animals. So this supports our hypothesis that the tocotrienol was boosting the antibody response to tetanus toxoid. And I think if you're interested, I can send you the 2 papers to which we reported the findings. Next, we talk about, again, how we've used the immune response. We use tocotrienol to enhance immune response to fight cancer. And the model that we've used is a syngeneic mouse model of breast cancer. Syngeneic means that the tumor induced comes from the same genetic background as the animal that is being induced. So that's an immunological term. So first study I'm going to share is the mouse model to assess if the anti-cancer effects of tocotrienol supplementation work through the T helper 1 and T regulatory cells. So the model was like this, about C, because that's the syngeneic mice, is about C. So we fed the mice with either gamma tocotrienol or vehicle, 0.5 milligram twice daily. And after 2 weeks, the mice was inoculated with the mouse breast cancer cells. And then we sacrificed a certain number of mice every 7 days for flow cytometry analysis, did some histopathology and immunohistochemistry analysis, and some proliferation and ELISA. I'm just going to share with you the IHC results and the flow cytometry results. Okay, this is showing you tumor growth in the different days. So here it's your tumor volume, and this one showing you tumor weight. And what you notice is the blue line, which is the gamma tocotrienol fat animals, is the size and the weight is significantly lower than that fat with vehicle. So we know something is happening in this animal to prevent the growth of the tumor. So then next we looked at the markers, the blood markers. So we looked at the different subsets of leukocytes in the blood. So this panel is showing you the proportion of CD4 cells in the blood on the different days. So what we notice is day 42, day 49, the number has increased, but we do not know which subset is increasing because you see T regulatory cells also has CD4. When we looked at CD8, okay, this is good because we saw that the CD8 T cells were increasing in the tocotrienol fat animals. Another anti-cancer leukocyte is natural killer cells. Again, we see that natural killer cells, although it's a little bit more up and down, we do see increase in the natural killer cells. Now, what was the most exciting is the T regulatory cells. what you see here is in the tocotrienol, gamma tocotrienol fat animals, the T regulatory cells were significantly lower. But in the vehicle animal, you see T regulatory cells were consistently high, suggesting to us that the Tregs in the vehicle fat animals may be suppressing the immune response. And in the gamma tocotrienol, maybe the tumor is not so big because the Treg function has been suppressed. So what we did, we made some sections from the tumor, okay, and we stained it with different antibody. I'm just going to show staining. So this is the tumor section from an animal fed with vehicle. So here, staining with CD4 antibody. See the cell, the antigen is present, okay, and you see there's some. But when you look at the FOXP3, which indicates FOXP3 is a transcription factor for T regulatory cells. What you notice is, totally, nearly the whole section was brown, which means there's a huge amount of FOXP3 there, suggesting immunosuppression. In contrast, when we looked at the tocotrienol fat animals, with the CD4, you still see a lot of infiltrate here. The CD4 is still high, but when we looked at the FOXP3 staining, the FOXP3 is actually very low. So this suggests maybe most of the CD4 cells that you're seeing here may not be FOXP3, judged by this FOXP3 staining. So that study suggested, yes, the immune system is being regulated and it's enhancing more of the Th1 response and to some extent suppressing the T regulatory function, allowing the body to fight the cancer, the immune system to fight the cancer. Now, the last study I'm going to share is actually the use of tocotrienol rich fraction as an adjuvant. Now, I mentioned already the dendritic cell, DC, dendritic cells are very important to boost immune response. So one of my PhD students had worked on using dendritic cell immunotherapy in the same model, and we showed there is a reduction in tumor growth. So what we wanted to do here is, if I also added tocotrienol rich fraction, will it make a difference? And I'm happy to tell you it did make a difference. So let me just share the study. So here the mouse is inoculated with the cancer cells, and tumor will be normally palpable about day 11 to 12. So what we did, we prepared dendritic cells from the animals, okay? And we pulsed the dendritic cell with tumor lysate. So we're creating an artificial situation like what would have happened inside the animal. So here the dendritic cell has been pulsed. And we also use control dendritic cell, which has not been pulsed. In one of the group, we fed them with TRF. So let me show you the graph, okay? So just to take you through, this is the control. This one here, the blue color is control. They receive the tumor, but no treatment. The pink, okay, the pink is they received the tumor and they were fed with vehicle. So it's good to see, the control and the vehicle, there were actually no difference. Okay, they're very similar. They behave very similar. The yellow bar is dendritic cell alone. Okay, that means no tumor lysate, no TRF. So you see the tumor growth, but it's not to the level of the control or the vehicle. The red bars is what we've done this previously in another study. We see a significant reduction in tumor growth. But the green bar is where we give DC plus tumor lysate, and we also supplement orally with tocotrienol rich fraction. You see the significant reduction in tumor growth. In fact, tumor growth is very, very small. And even at week 11, you see very, very small tumor. So again, the criticism here would have been, tocotrienol may be acting directly on the cancer, killing the cancer cell. So we wanted to see, was this reduction due to some changes in the immune response? So we took the peripheral blood from these animals and isolated CD8 expressing T cells. If you remember, CD8 T cells are the ones that actually kill the tumor. And we incubated the CD8 T cells with the cancer cells at different concentration. So what you see here, this is the ratio is 5:1, 10:1, 15:1, 20:1. There's a ratio of the cytotoxic T cell to the cancer cell. Now, immediately you can see the blue lines and the pink line, which is the vehicle and the control. And this is the DC alone. You don't see a big difference. The red line is showing you cytotoxic T cells from the DC plus tumor lysate group, which you see there is some significant killing. And the green line is the cytotoxic T cell isolated from the DC plus tumor lysate plus TRF. Here it shows you that we have generated tumor-specific CD8 T cells because we see killing of the cancer cells in this model. So this was published in this journal. Okay. So just to conclude, we've shown that TRF supplementation, I showed you the evidence, TRF supplementation enhanced vaccine-specific antibody production in a clinical trial with human as well as in a mouse model. I shared the evidence. And in the mouse model of breast cancer, we showed that it modulated the host immune system, allowing the host immune system to fight the cancer, as well as the supplementation of tocotrienol rich fraction acted as an adjuvant to promote DC, dendritic cell-based immunotherapy in breast cancer. So I would like to conclude the session by reiterating that tocotrienol supplementation can enhance host immune system. Thank you. So if any of you want the publication, the PDF, please ask Angeline, and I'm happy to share this with you.
Thank you so much for the informative session, Prof Ammu. So we've actually received a couple of questions. So maybe let's take our time, a couple of minutes to just, get you to answer some of the Q&As that we've received. So the first one is, which is stronger for immune system regulation, vitamin C or vitamin E?
Okay, it's difficult to answer that question because there's not been a study where we did a comparison between vitamin C and vitamin E. But what I can say is they're both antioxidants. So they may be working similarly, but until you do a study and see, okay, this is working this way, this is working that way, it's difficult to say which is better. Both are antioxidants. So maybe as antioxidants, both are effective.
Perhaps just a follow-up question that I have from there, right? Given that one is oil soluble and one is water soluble, do they work sort of differently to sort of regulate?
Okay, the oil soluble. Okay, that's a good question. Good question. Yeah, vitamin E is a lipid soluble vitamin, so it can actually stay in your cells. It can stay in your fat tissue for longer. Vitamin C is a water soluble, so any excess is actually thrown out, goes out of your body. So maybe in that sense you could say vitamin E is better because it stays there. It can be stored in your tissues. Yeah.
Great. Yeah, thanks for that. So I suppose linking the next question is, what is the recommended dosage of tocotrienols proven to support or boost the immune system?
Okay, very, very interesting question. Okay, as I shared with you, the trial, we use 400 milligram. Okay, so we need to do another trial where we use lower doses of vitamin E, okay? Because we know now we don't have to go more than 400 because 400 works. So if you want to ask me the minimum, we still need to do the study. And I think we are planning that study now to do lower doses to see whether we get, which is the lowest dose that you need to take to get a response. So okay. Up to 4. So I can't answer that question because we haven't done the research.
Right. No worries. So everything that's been sort of recommended has to be based on the research. Studies that you've conducted, right?
Yes.
So the next question,
or even other people's studies. So other people also have not done the study, so difficult to comment.
Okay. In that case, let's move on to the third question. What are the considerations for taking tocotrienols before getting vaccinated?
What do you mean? Should you take, or what are the, actually vitamin E is a supplement. It's a natural product. It's found in the oil, especially palm oil. So it's a natural product. So what are you asking me, how long you should take it, or is it recommended to take? I'm not understanding the question.
Okay, so maybe we'll wait for any follow-up comments from there.
So just to tell you, in that study, in the mouse model, we gave 2 weeks supplementation before the vaccination. In the human study, we gave 4 weeks supplementation before the vaccination. So both we saw protection. I suppose even if 1 week, it will give you protection. I mean, that's what I would predict. But again, you need to do, before you go and put your neck out, you've got to do the research.
Okay, all right. Okay, so I think there are a couple more questions that are coming in now as we speak. So the next one is, from Ahmad Zahidi. The question is, what is the effect for people who take methotrexate on the immune response by tocotrienols?
Methotrexate. Okay. Again, not done, but I know there's a study in the US where they showed that taking tocotrienols before you go for again, this is animal model, before you go for radiation, it seems to protect your bone marrow derived cells, your immune cells, your red blood cells. By that token, methotrexate actually is an anti-proliferative agent, so it may cause a problem in your proliferation, because methotrexate will stop T cells from being, when your T cells are activated, they need to proliferate, and then they will differentiate into your memory cells and whatever cells are making cytokines or plasma cells making antibody. So methotrexate may interfere in that aspect. Yeah. So there could be a problem, but as I know, in many conditions taking vitamin E, we have not seen any adverse effects.
Fantastic. Okay, so coming from Sabrina, question is whether can cancer patients that undergo chemotherapy take tocotrienols?
Okay, so that's an interesting question. I mean, I'm not a medical doctor, but what I can, as I said to you, the going through like radiation this is an American study which showed that if you feed the animals with tocotrienol and then you put them through some radiation thingy, it seems to protect their immune system. Okay, so chemotherapy, but chemotherapy actually attacks a rapidly dividing cells. We don't know whether it will protect, but what we know is in some cancer patients, which we have done some initial trial, again, breast cancer patient, we actually saw their red blood cells coming back faster.
Very interesting insights.
Interesting, but again, these kind of questions, until you do the research, you don't want to give an answer, because it would look like you're saying you're hitting in the dark. But if you have the evidence, it's easier to say, yeah, you can do this, yes, you can do that. So what I'm trying to say is there's a lot more research to be done.
Yeah, but the preliminary type of evidence shows, for it to be positive in this case. Yeah. Okay. So the next question from Junying Woon is whether a patient taking vitamin E to prevent can a patient take vitamin E to prevent the further growth of cancerous cells after being diagnosed with cancer?
Okay, a lot of cell-based experiments using different cancer drugs in the cell-based condition have shown that most drugs work synergistically with tocotrienol. Okay, so again, it's difficult a lot of clinicians are worried about using like supplements, which it may interfere with their drugs. So again, we need to do studies. I think treatment-wise, it's a bit more difficult for me to answer. I think what I'm trying to show you is it's prevention. It's using tocotrienol to prevent you going that way. So you have tocotrienol, you boost your immune system is healthy, your whole body is healthy, so it can fight things better. So as a treatment, it will actually take a lot more work. We actually need to do a lot more research to make claims. And you cannot make claims without evidence. So, but what I'm claiming now is take it, it's health, keeps you healthy. So I'm talking more about healthy people, remaining healthy.
Yeah. I think a lot of there's a lot of interest in cancer just because there's so much sort of uncertainty.
So you take the tocotrienol, keep your immune system good and vigilant. So one thing about the immune system is it doesn't like not doing something. So there is a theory in the literature called hygiene hypothesis. So nowadays there are a lot more people presenting with allergy and asthma, and that is a Th2 response. This is because a lot of us, very clean, don't want to expose to any pathogen, don't want to give your immune system any work. So when that situation happens, the immune system now attack things that are not harmless, like pollen, dust. I mean, what can that do to you? So you find that people are developing more of the allergy response. So the hygiene hypothesis says you must regularly challenge your immune system. And that's why people who get more vaccination regularly, you find that they actually fight better. So there is a paper that I shared with you, remember, the influenza vaccine. People who take influenza vaccine seem to have a milder form of COVID-19. So that supports my theory, hypothesis. So you need to keep your immune system, working, happy.
Yeah, challenge it, give it the growth that it needs, right? In the right way.
Give it a little challenge now and then. So then it's working. It's not
right. So I think, now we're nearing the end. We've got one question, and if anyone has more, now's the time to type it in. So question from Heng Yi Xian is that you mentioned that tocotrienols can be used as a neuroprotector. Can it be used to reduce cardiovascular diseases as well?
Okay, the neuroprotector I can just share with you, there is a clinical trial happening because I've done work with cell lines and mouse, rat models of Parkinson's. We saw protection. There is now a clinical trial happening, I think in Singapore, on the use of tocotrienol to protect Parkinson's. So CVD, yes, again, you look at the literature, it shows you that cholesterol can be reduced. The tocotrienol intake actually reduces cholesterol and gives you the good cholesterol. So that could answer the CVD. And another thing, maybe I should also tell you is tocotrienol works quite similarly to aspirin. It's like a blood thinner. So so in that sense, it probably is good for all these conditions.
Yeah, I think now we're nearing, we've reached the end of today's session. So it's 3 o'clock now. So if there are no more questions that are coming in, I mean, if there's any, you can just let us know as well, and we'll definitely get in touch with Prof Ammu to ask her if she's okay with that. And at this point, thank you all so much again for attending our webinar today. Thank you, Prof, for such an enlightening session. I think all of us learned a lot today. This word immunity or immune system is always used on a daily basis now, right? And I think today we've really got that deep insight on. Well, I think we're just scratching the surface, but for me today, it's really deep learning. And thank you so much again on behalf of Sime Darby Oils Nutrition. And for our attendees today, if you're a member of the Malaysian Pharmacists Society, you would be eligible for 1 point for attending today's session. So do reach out to us if you have any questions. Meanwhile, may the rest of you have an awesome rest of the day. Thanks for spending your afternoon with us, and we can't wait to see you soon for our next session.
Okay, thank you so much.
Bye, everyone.
Bye-bye.